So everybody is able to make over 10 trillion different antibodies, which is really crazy to think about. So how this happens is that we rearrange the antibody gene segments in the DNA when we're making the B lymphocytes. There's also some variations in the RNA splicing, and also the B lymphocytes have a high rate of mutation. So how we're able to produce so many different B lymphocytes that recognize lots and lots of different pathogens is that in the stem cells in the bone marrow, so as they're making the new B lymphocytes, they just randomly cut out parts of the DNA that code for the receptors on the B lymphocytes. So it's just random what kind of receptor they're going to make. And so we have to be ready for everything because we don't know what we're going to be running into. So we've got millions and millions of different B lymphocytes that recognize different antigens. And of course, some of them do recognize body parts, so we do need to get rid of those. Once the antibodies are made by the plasma cells, there's several ways that they can work to help protect us. So just one vocabulary word is an immune complex or antigen-antibody complex. So this picture here is just showing you an immune complex. So it's just the antibody bound to the antigen, and that's just called an immune complex. So let's take a look at the different ways that the antibodies work. So one way that they can work is through neutralization. So when you have antibodies binding, for example, to the spike proteins on the virus, that's going to block the virus from binding to receptors on our cells, and so they're not able to get inside. And the same thing with the bacteria and also toxins. Another way antibodies work is they cause agglutination. Agglutinate just means to clump. So when the antibodies bind to these bacteria, for example, because you've got two antigen binding sites on each end of the antibody, you can actually bind to two bacteria at once. And so you can end up with these big clumps of bacteria, and that will trap the bacteria, and then they're not able to spread to other places in the body. And it also makes it easier for the phagocytes to grab onto the bacteria when they're all clumped together like that. So that's agglutination. Another way that antibodies work is, remember, they are absonins. So they can coat the bacteria and make it easier for the phagocytes to grab onto them and eat them. And remember the other absonins? Those would also be complement proteins and acute phase proteins. And then antibodies can also activate the complement system. So if the antibodies are bound to this bacterial cell, that will set off the first complement protein, which will set off the next, and you set off that cascade. And here we're showing you the end result of the complement system forming this membrane attack complex. Remember, the water will pour in there, and then the bacteria explode. And another thing that antibodies do is they help identify cells that are abnormal, either infected or cancerous, and the natural killer cells. Remember, that's one of the ways that they recognize that a body cell is abnormal. So if it's coated with antibodies, that's like a big red flag, and that'll stimulate the natural killer cell to kill that cell. And then the other way that the natural killer cells will kill cells is, remember, if they are missing their ID tag. So if they don't have a class I MHC protein, that's another signal for the natural killer cell to kill the cell.